Hi guys, welcome to the discussion of Chapter 6, Expression of Biological Information. In this video, we are going to be focusing on gene regulation and expression by looking at operon as an example. These are the learning objectives that you need to complete by the end of the lesson. At the end of the lesson, students should be able to explain the concept of operon and gene regulation. State the components of operon. The students should also be able to explain the components of operon and their functions in E. coli. And lastly, students need to be able to explain the mechanism of operon in the absence and presence of lactose. Do you still remember in the previous video that I mentioned transcription occurs throughout the life of the cell? Transcription of certain genes will happen as the cell needs the product coded by the gene. If the product coded by the gene is no longer needed by the cell will prevent transcription of that gene from happening. So therefore, from here, we can say that gene is regulated, or in other words, being turned on or turned off by allowing or preventing transcription from happening. How does the cell does this? The cell does this by allowing or blocking access of RNA polymerase, i.e. the enzyme that's responsible to carry out transcription, to the promoter of the gene. To learn about the regulation of genes, or in other words, how genes are turned on or turned off, we will look at operon that is present in E. coli. What is operon? Operon is a functional unit of DNA made up of a cluster of genes, all under the control of a single promoter. For example, like operon, we have the gene Z, Y, and A that individually code for protein, hence we call them as structural genes, and all these genes are under a control of a single promoter. The Z gene, Y, and A all codes for products which are all involved in lactose metabolism in E. coli. It is important for students to note that operon can only be found in prokaryotic cells. The genes in eukaryotic cells are not organized into operons. Now let's have a look at component of operon. We have structural genes consisting of Z, Y, and A, all codes for protein that are involved in lactose metabolism. Z is the gene code for the enzyme beta-galactosidase. This enzyme breaks down lactose into glucose and galactose. Y is the gene that codes for lactose permease. This is the transport protein that pumps lactose into the cytoplasm of E. coli. A is the gene that codes for transacetylase. Transacetylase is an enzyme that helps transfer the acetyl group from acetyl-CoA to galactosides, lactosides, and glucosides. These are all intermediates that is present during lactose metabolism. Besides structural genes, we have the operator. Operator is the site of binding for repressor protein. Repressor protein is coded by a regulatory gene. This gene can be found upstream from the operon. The regulatory gene is always on. So therefore, the repressor protein is always present in the cytoplasm of the cell. This is how the repressor protein binds to the operator. Next, we have the promoter. Promoter is the site of binding for RNA polymerase. RNA polymerase will bind to the promoter of the operon, unwind the DNA, and begin to move downstream, carrying out the transcription process, resulting in the production of mRNA. The translation of the mRNA will result in the production of the various proteins that we have just mentioned. Now let's have a look what happens to the operon in the absence of lactose. As I have mentioned earlier on, the regulatory gene is always on, thus causing the repressor protein to always be present inside the cytoplasm of an E. coli. In the absence of lactose, the repressor protein are able to bind to the operator of the LAC operon. This prevents the RNA polymerase from binding to the promoter of the LAC operon. Since RNA polymerase is prevented from binding to the promoter of the LAC operon, the transcription of the structural genes cannot take place. So therefore, no mRNA is produced, hence, no protein will be produced. In this case, the LAC operon is said to be turned off. How does the cell turn on the LAC operon? The LAC operon will be turned on in the presence of lactose. Lactose will be converted into allolactose. The allolactose will then bind to the allosteric site of the repressor protein. This causes the repressor protein to change its shape. Hence, it's no longer fit to the operator. This causes the repressor protein to detach itself from the operator. This then allows the binding of RNA polymerase to the promoter, resulting in the transcription of structural genes producing the mRNA. Translation of the mRNA will result in the production of the various proteins that we have discussed. These proteins will then help E. coli cell to carry out lactose metabolism. In this case, LAC operon is said to be turned on.